Welcome back to the deep dive. Today, we are going to look at a scenario that is, well, it's basically the absolute nightmare of every drug developer.
It really is. It's the scenario that keeps chief medical officers awake at night.
Exactly. Imagine this. You have discovered a molecule. In the test tube, it's perfect. In the initial efficacy models, let's say it shrinks a tumor or clears a nasty infection, it works like a charm.
Looks like a miracle cure.
Totally. Investors are happy, scientists are popping champagne, but then you give it to a living creature for the first time
And it stops their heart.
or it shuts down their breathing, or sends them into a massive seizure.
Right.
And just like that, you don't have a medicine anymore, you have a poison. That razor-thin line between a chemical and a medicine is exactly what we're exploring today with the ICH S7A guideline.
It's a dramatic way to put it, but you're absolutely right. This is the gatekeeper. The document is officially titled Safety Pharmacology Studies for Human Pharmaceuticals.
Or just S7A.
Or just S7A if you're in the industry. And honestly, if you're a student preparing for an exam on drug development, toxicology, or regulatory affairs, you cannot afford to skim this. This document is foundational.
So for everyone listening, whether you're cramming for that final or you're a professional trying to understand how we actually keep patients safe in phase 1, we're going to break this down so you can ace the test.
Yep.
We have the final version of the document right here, dated November 2000. It's a tripartite guideline. Now, before we get into the biology, what does tripartite actually signal to the students listening?
It signals global consensus. It means this wasn't just written by the FDA in a basement somewhere.
Right.
It was harmonized between the European Union, Japan, and the United States. So the rules we're discussing today are the global standard. If you want to develop a drug for the major markets, you follow S7A.
And what's the actual mission statement here? Because usually we think of testing as just seeing if the drug works.
Right, but S7A has this dual mission. First and foremost, protect clinical trial participants. We need to make sure the first human volunteer isn't put at unreasonable risk.
Of course.
But the second mission is interesting. It's to prevent the unnecessary use of animals. The idea is to have a testing strategy that's smart and targeted, so we aren't just testing everything on everything for no reason.
So we want the maximum data from the minimum number of animals.
Precisely. Smart and targeted.
I like that. Let's get right into the definitions, because I know for a fact this shows up on exams. If I'm a student and the question asks for the textbook definition of safety pharmacology according to S7A, what am I writing down?
Okay, get your pens ready. Safety pharmacology is defined as the investigation of the potentially undesirable pharmacodynamic effects of a substance on physiological functions.
Okay, let's unpack this. Undesirable pharmacodynamic effects. That sounds a lot like toxicology.
Yeah.
Or even just side effects. How do we distinguish safety pharm from toxicology?
That is the critical distinction. It's where people get tripped up.
Or from primary pharmacodynamics?
Right. So think of a Venn diagram. Primary pharmacodynamics is what you want the drug to do. It's the mechanism of action binding to the tumor receptor, lowering cholesterol. That's the benefit side.
Okay, so primary PD is the goal.
Exactly. Now, toxicology, generally speaking, looks for structural damage. It's looking for pathology.
Like, did the liver cells die?
Exactly. Did the kidney tissue get inflamed? It's looking for hardware damage, and usually that happens after repeated dosing over time.
So where does safety pharm fit in all this?
Safety pharm is this unique middle ground. It's looking for functional problems, not structural ones. It's looking at the software.
Ooh, I like that.
Did the heart rhythm glitch? Did the blood pressure spike? And crucially, it investigates these effects within the therapeutic dose range and slightly above. We aren't looking for what happens if you massively overdose. We're looking for what might happen when you take the medicine as intended.
That's a great analogy. So toxicology is, did the engine block crack? And safety pharm is, did the accelerator get stuck?
Precisely. And because these are functional changes, like a missed heartbeat or a drop in oxygen, they happen acutely, they happen fast. So safety pharm is all about real-time physiological observation. You give the drug, and you watch the system react, in the moment.
This brings us right to the core structure of the guideline. Because S7A doesn't just say, check every organ system. It actually creates a hierarchy of the body.
It does.
It prioritizes systems based on how critical they are for keeping you alive right this second.
Correct. We call this the concept of vitals. The guideline separates body systems into the core battery, the absolute essentials, and then the supplemental systems.
Let's focus on that core battery first. I call these the big three. If you were taking notes, this is probably the most important part of our whole deep dive.
It really is.
Central nervous system, cardiovascular system, and respiratory system. Why these three? Why not the liver? Why not the kidneys?
Well, think about the immediate consequence. If your kidneys stop working for an hour, it's not good. But you aren't going to die right there on the table.
You've time to intervene.
You do. Dialysis exists. But if your heart stops pumping for 5 minutes, if you stop breathing, if your brain goes into status epilepticus, that's the end game. Immediate death or irreversible harm. That's why S7A mandates that these 3 must be tested before a drug ever goes into a human.
Okay, let's break them down one by one, starting with the central nervous system, or CNS. We obviously can't ask a rat if it's feeling dizzy or depressed, so what are we actually measuring here?
We're looking for observable functional changes. The guideline lists things like motor activity, is it moving too much or too little? Behavioral changes, coordination, sensory reflexes, and even body temperature. But you can't just casually glance at the animal.
No, it has to be standardized. The document mentions a couple of specific tests here. The Functional Observation Battery, or FOB.
And Irwin's test, right? So if you see Irwin's test on an exam, immediately link that to CNS safety pharmacology. These are systematic protocols. An observer goes through a checklist. Do the pupils constrict to light? Does the animal grip the cage or slide off? Is it walking with a weird gait?
So it turns subjective observation into objective data.
Exactly.
Okay, next up is the cardiovascular system, CVS. This seems obvious. Blood pressure and heart rate.
Yes, but there's a nuance in the text that students often miss, and it's a huge one. It is not just blood pressure and heart rate. You must evaluate the electrocardiogram, the ECG, and specifically the guideline notes the importance of looking for repolarization and conductance abnormalities.
Repolarization abnormalities. That's code for QT prolongation, isn't it?
It is, and this is vital context. This guideline came out in 2000. Around that time and in the years prior, several non-cardiac drugs, antihistamines, antibiotics, were being pulled from the market because they were causing sudden cardiac death.
The silent killer.
Exactly. The drug didn't change the heart rate, it didn't change the blood pressure, but it delayed the electrical resetting of the heart, the repolarization. If that reset takes too long, the next beat can trigger a chaotic rhythm called torsade de pointe.
Which is often fatal.
S7A was really forward-thinking in locking this down. You have to look at the waveforms, not just the beat count.
So it's looking for electrical instability, not just mechanical failure.
That is a perfect way to describe it.
Got it. So look at the ECG and the third one, the respiratory system. This is where I think a lot of people get tripped up. I feel like the assumption is, well, the animal is alive, so it must be breathing.
That is a guaranteed exam trap. The guideline specifically states that clinical observation of animals is generally not adequate.
So you can't just look at the cage.
Nope. You can't just say, yep, chest is moving. You need numbers.
Quantitative measurement.
You need quantitative measurements. Respiratory rate, tidal volume, which is how deep the breath is, and often oxygen saturation. Think about opiates. An animal on an opiate might breathe at a normal rate, but the breaths become very shallow.
You'd miss it if you're just watching.
You might. But if you measure tidal volume, you see the crash in oxygen exchange. Safety Farm catches that hidden depression.
So that's the core battery, CNS, CVS, respiratory, the big three. But let's say we run the core battery and we see something. The CNS test shows the animal is acting confused or there's a tremor. What happens next?
That triggers what S7A calls follow-up studies. You've identified a red flag in the core battery, so now you need to dig deeper into that specific system.
Can you give us an example?
Sure. If your core CNS test showed, say, weird behavior, a follow-up study might specifically test learning and memory, or visual and auditory function. Or if your core cardiovascular test showed drop in blood pressure, a follow-up study might look at cardiac output or ventricular contractility to understand why the pressure dropped. Is the heart weak, or did the blood vessels dilate?
So follow-up studies are deep dives into the big three when things go wrong, but there's another category called supplemental studies. How is that different?
Supplemental studies look at these second-tier organ systems. These are systems where damage is bad, but it's usually not immediately fatal in the acute setting. We're talking about the renal urinary system, the autonomic nervous system, and the gastrointestinal system.
I really like the car analogy you used when we were prepping. Can we bring that back to explain the difference between core and supplemental?
Oh, absolutely. It's the best way to visualize the hierarchy. The core, battery, CNS, CVS, respiratory is like checking the engine, the brakes, and the steering. If any of those fail while you're driving down the highway at 60 miles per hour,
You crash immediately.
you crash. You have no control. The supplemental studies are like checking the air conditioning, the stereo, or the windshield wipers. You need them for the car to function properly in the long run. If the
Right.
AC breaks, it's uncomfortable. If the wipers fail, it's dangerous, but you can usually pull over.
But the car won't immediately veer off the road and explode.
Exactly. That puts it perfectly.
So for supplemental studies, say, for the renal system, what are we looking for?
You would check things like urine volume, specific gravity, osmolality. For the GI tract, you might look at gastric secretion or transit time. Does the drug make things move too fast or too slow? Again, these are important, but S7A prioritizes them lower than the vital functions.
Unless there's a specific reason to be worried based on the drug's mechanism.
Exactly. Vitals first, everything else second.
That makes the hierarchy really clear.
Yeah.
Now let's shift gears to practical application. We know what to test, but how do we design the study? The guideline is pretty strict about the timing.
This is a hard stop in drug development. The safety pharmacology core battery must be completed prior to the first administration in humans.
Full stop.
Full stop. You cannot start a phase 1 clinical trial until you have this data. It's a regulatory gate.
And what about the animals themselves? I imagine in a lab setting, it's easier to work with an anesthetized animal. They're easy to monitor. But what does S7A say?
S7A actually has a very strong preference for unanesthetized conscious animals.
Why is that? Wouldn't anesthesia reduce the noise in the data?
It reduces noise, but it also blunts the signal. Anesthesia fundamentally alters physiology. It lowers blood pressure, it changes heart rate, it depresses respiration.
So you could get a false negative.
You could. Imagine you're testing a stimulant like a new caffeine derivative, but you test it on an animal that is heavily sedated. You might not see the heart rate spike because the anesthesia is suppressing it.
And a false negative is the most dangerous thing in safety testing.
It is. You tell the FDA it's safe, but it's not. The guideline actually says data from unrestrained animals using telemetry, where you implant a transmitter and let the animal walk around freely, is the gold standard.
Because stress also changes physiology.
Exactly. You want the baseline to be a happy, normal animal.
Now let's talk dosing. In efficacy studies, you use the therapeutic dose. Is that enough for safety farm?
No, and this is another critical exam detail. You must test the therapeutic range and above. You are trying to define the margin of safety.
You're looking for the cliff edge.
Right. If the effective dose is 10 milligrams, but the heart stops at 12 milligrams, you have a huge problem. That drug is undevelopable. You need to push the dose until you see adverse effects or until you reach a reasonable limit. You need to be able to tell the clinicians you have a safety window of 50-fold, or be careful, the window is only 5-fold.
You need to see the dose response of the bad effect.
Precisely. And you need negative and positive controls to prove your test system actually works.
Right.
If you give a drug known to cause arrhythmias, your positive control, and your system doesn't pick it up, your safety data for the new drug is worthless.
Makes sense. Now, I want to touch on something that confuses students regarding good laboratory practice, or GLP. Does Safety Farm have to be GLP?
This is a nuanced answer, so listen carefully. The core battery, remember, the big three must generally be GLP compliant.
So audits, rigorous documentation, all that.
All of it. This is because it's a pivotal safety study that supports human life. But the efficacy studies?
They don't have to be GLP.
Generally, no. Primary pharmacodynamics, efficacy stuff, does not need to be GLP. And typically, secondary pharmacodynamics or exploratory studies don't need to be GLP either. But when you are submitting that core battery data to the FDA or EMA to open a clinical trial, that needs to be the highest quality data possible.
Are there any exceptions? Are there times when you can just skip Safety Farm entirely?
There are a few specific scenarios. First, locally applied agents. If you have a skin cream or eye drops and you can prove the drug doesn't get into the blood,
Low systemic absorption.
right, then you might not need a full systemic Safety Farm battery because it's not reaching the heart or brain to cause trouble.
Makes sense. What else?
Another exception has been cytotoxic agents for end-stage cancer. The logic was that these patients are in a desperate situation and the drugs are known to be toxic anyway, they kill cells, so we accept more risk. The guideline says studies prior to the first human dose might not be needed.
But that's changing a bit, isn't it?
It is. The guideline notes that for novel mechanisms, even in cancer, it might still be valuable. But strictly speaking, for classic cytotoxics, there's an allowance there.
And what about biologics? Monoclonal antibodies.
Good question. For biotechnology products that have highly specific receptor targeting like an antibody that only hits one protein on one type of cell, S7A says you can often evaluate safety endpoints as part of your general tox studies. You might not need a separate standalone safety farm study.
Because the off-target effects are much less likely.
Exactly. Much less likely than with a small chemical molecule that might float around hitting 5 different receptors.
One last technical detail before we summarize. Metabolites.
Ah, yes. The human metabolite issue. This is a classic curveball.
Explain it to us.
So sometimes a drug enters the human body and the liver chops it up into a new molecule, a metabolite that is unique to humans. Or maybe it's present at much higher levels in humans than in the rats or dogs we tested.
So the animal testing didn't actually cover that specific molecule.
Exactly, you have a blind spot. In that case, S7A says you need to test that metabolite separately. You can't just assume it's safe because the parent drug was safe in the dog.
Okay, we have covered a massive amount of ground. From the definition to the hierarchy, the specific tests, study design, the exceptions. Let's synthesize this. I want to give our listeners the cheat sheet.
Let's do it.
If you are walking into the exam hall in 10 minutes, what are the absolute essentials you must remember?
Let's do the rapid-fire summary. Here are the 6 things you must know.
Number 1, the definition.
Safety pharmacology investigates undesirable pharmacodynamic effects on physiological functions. It's distinct from primary PD and from toxicology.
Number 2, the core battery.
The CNS, cardiovascular, and respiratory systems. The vital systems. Acute failure equals immediate death.
Number 3,
The timing.
must be completed before the first administration in humans.
Yeah.
No exceptions for the core battery.
Number 4, the methodology.
Strong preference for unanesthetized conscious animals. Anesthesia masks physiological responses.
Number 5, the dosing. You must test the therapeutic range and above. You have to define the safety margin.
And number 6, GLP status.
The core battery requires GLP compliance. Primary pharmacodynamics generally do not.
There you have it. That is your roadmap to S7A.
You know, before we sign off, I think it's important to reflect on why this all matters. It's easy to get lost in the alphabet soup, CNS, CVS, GLP, FOB.
This is.
But safety pharmacology is really the bridge. It's the bridge between a chemical in a flask and a medicine in a human.
It's the safety net.
It is. Standard toxicology looks at tissue slices and blood chemistry. It catches liver failure. It catches kidney necrosis. But standard toxicology can miss the silent killers.
Right.
A rat in a toxicity study might look fine, but if its QT interval is prolonged, you wouldn't know until you measured it with an ECG. Safety Pharm is designed to catch those invisible functional glitches that don't leave a mark on the tissue but can stop a heart.
It turns drug development from a gamble into a science.
That's a powerful way to look at it.
It isn't just a regulatory checkbox, it's a life-saving discipline. It's the reason we can volunteer for clinical trials with some degree of confidence.
Absolutely. It's the unsung hero of the IND package.
All right, everyone, take a deep breath. Review that list of the core battery one last time. CNS, CVS, respiratory, remember the car analogy engine, brakes, steering. You are going to do great on the exam.
And remember, we are discussing the S7A document specifically. Regulations evolve, but this is the foundation.
Thanks for listening to this deep dive. Go ace that test.